Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Isolating CD4 positive and CD8 positive T cells for improved drug efficacy and increased productivity. I'm Caitlin Crouch, and I'll be moderating today's webinar. And joining me today is Haley Mios, who will explore a method that enables isolation of CD4 positive and CD8 positive T cells with a key differentiator, an active release mechanism for clinical trial or commercial manufacturing. After our presentation today, we'll have a live Q&A session. So please do feel free to pose your questions to our speaker using the Ask a Question box at the bottom of your screen, and we'll try to get to them during today's session. I'd also like to draw your attention to the Resources tab on the right, where you can find more information on the topics covered today. Now, I'd like to introduce today's presenter. Haney is an R&D staff scientist at Thermo Fisher Scientific and the project lead for the CTS detachable dynabeads and the CTS DynaSelect magnetic separation system. Haney obtained a bachelor's degree in molecular biology from the University of Sheffield in the UK and a master's and PhD in molecular medicine from the Norwegian University of Science and Technology in Norway. So, without further ado, I'll hand it over to Haney to kick us off with today's presentation.
Thank you, Caitlin, for the introduction. It is my pleasure to talk to you today about the Thermo Fisher vision for cell therapy manufacturing and our two new offerings from our detachable dynabeads product line, the Gibco CTS detachable dynabeads CD4 and CD8. In Thermo Fisher Scientific, we place our customer in the center of all of what we do. We focus in helping our customers to accelerate innovation and enhance productivity. We partner with our customers to speed up routes to market by providing support across many levels, from process optimization to regulatory compliance. We in R&D regularly engage with our customers and collaborators, giving them access to our know-how and experience acquired during product development. And we take with us their feedback and pain points to inform our innovation roadmap. We understand the challenges that our customers face when developing cell therapy products. Today, manufacturing challenges in cell therapy continually focus on ensuring patient safety and reducing cost to expand treatment access to patients. So to address these issues, automation and closed operation during manufacturing increases efficiency, decreases variability, and reduces the risk of contamination. Also, as the field progresses, allogeneic and autologous products will most likely coexist, though a flexible and a scalable platform is needed to meet cell therapy manufacturing at a range of scales. Our next generation platform of CTS detachable dynabeads are designed to address the challenges that I just talked about. So building upon the clinical track record of our previous CTS dynabeads, this new family of products possesses an active release mechanism that is the first to exist for process development, clinical trials, and commercial manufacturing. The active release mechanism allows the users to actively detach the beads from the cells at any time within the process, though giving the user fine control over the manufacturing process. And the CTS detachable dynabeads CD3 CD28 kit was the first product to launch in this product family. But today, I'll talk to you about two new products, the CTS detachable dynabeads CD4 kit and the CTS detachable dynabeads CD8 kit. The beads are designed for the isolation of CD4 and CD8 T cells, as the name suggests. The detachable mechanism allows for the removal of the beads following isolation. And finally, the new platform of CTS detachable dynabeads is optimized for the use with the CTS DynaSelect magnetic separation system, which I will talk to you about in detail shortly. So the result is an automated, closed system, flexible, and a scalable solution. In addition to the active release, the CTS detachable dynabeads utilize our capture select technology. The beads are coated with single domain VHH nanobodies that are derived from llamas. The single domain VHHs are about 15 kilodalton and contain the C-terminal variable regions that recognize antigen. So single VHHs have lower binding affinity when released from the beads, though allowing them to simply detach from the target cells after release of the beads, and then they can be easily washed away. We produce the VHHs in yeast, so the product is animal origin free and excludes any risk of viral-like particle contamination that could be an issue with conventional antibodies produced in hybridomas. I will now walk you through how the CTS detachable dynabeads technology works. This process that I will be talking about is fully automated on the CTS DynaSelect magnetic separation system. So the CTS detachable dynabeads are about 4.5 micron paramagnetic polystyrene beads, similar to our legacy passive detached CTS dynabeads that are well known, but with two main differences. One is the active release technology, and two is that they are coated with VHHs instead of conventional antibodies. So during isolation, the beads are incubated with material containing mixtures of cells, such as leukapheresis, for example, and then the beads will bind to the target cells, and now the bead-bound cells can be retained on the DynaSelect magnet, and the non-target cells can be washed away. When the provided release buffer is then added to the bead-bound cells, biotin, which is the active ingredient, interacts with the release mechanism, resulting in the detachment of the beads from the target cells. And then using the DynaSelect magnet, the cell-free beads can now be captured, and the VHHs and the release buffer can now be washed away from the cells, and the highly pure target cells are now ready for downstream application. So this new breakthrough platform of products includes catalog offerings for CD3/CD28 for one-step isolation activation of T cells that was recently launched, and the two new catalog products for the isolation of CD4 and CD8 T cells that will expected to launch in early 2024. So I will now play a short cinematic video that shows how the active release mechanism works.
The Gibco CTS DynaSelect magnetic separation system is a closed, automated device for cell isolation and magnetic removal of dynabeads. It also acts as the foundation for Thermo Fisher's next-generation platform, the Gibco CTS detachable dynabeads. This breakthrough offering is the first to have an active release mechanism for clinical trial and commercial manufacturing use. Coated with camelid-derived single-domain VHH antibodies, detachable dynabeads demonstrate high specificity as they bind to target cell surface markers. An accompanying release buffer competitively displaces a biotin derivative on the surface of the dynabeads to allow an active detachment from target cells at any time. The beads are then magnetically removed, resulting in a highly pure cell population. With this active release mechanism, users are empowered with process flexibility characterized by high purity, favorable yield, and desired cell phenotype. The new wave of detachable dynabeads will serve autologous and allogeneic processes to the benefit of cell therapy patients.
Okay, now I will talk a bit about the CTS DynaSelect magnetic separation system, which is the foundation for the next generation platform of CTS detachable dynabeads. DynaSelect is an instrument for automated magnetic cell separation and bead removal, and it is intended to be used with all of our detachable beads. The instrument is accommodated by single-use, fit-for-purpose kits for isolation and bead removal. And the key feature of the instrument is an automated magnet. The magnet is situated on a rocker, which enables a mixing of cells and beads during cell bead incubations. Automated fluid management is achieved by a peristaltic pump and pinch valves, allowing the transfer of the fluid between bags. Fluid movement and pressure is monitored by bubble sensors and pressure occlusion sensors, respectively. The instrument has embedded graphical user interface with a customizable software that enables flexibility from process development to commercial manufacturing. And the DynaSelect software is 21 CFR Part 11 compliant. The instrument can be used as a standalone or connected to other instruments as a part of a workflow. This is enabled by open platform communication, unified architecture, and distributed control systems such as Emerson Delta V. In addition to DynaSelect, cell therapy systems instruments include the GIBCO CTS Rotea counterflow centrifugation system and the GIBCO CTS Xenon electroporation system. All of our CTS instruments can be used as a standalone or integrated physically and digitally to create a modular end-to-end workflow controlled by Emerson Delta V for complete automation. All of the Thermo Fisher cell therapy systems products guarantee GMP compliance and come with regulatory documentation and support. Now I will focus on our new products. And first, I will talk to you about the CTS detachable dynabeads CD4 kit. So this product is intended for isolation of CD4 T cells. And I will now share with you isolation performance data that was generated on the CTS DynaSelect magnetic separation system. So here is a summary of the protocol details. The starting material is cryopreserved leukapheresis from healthy donors. And these runs were conducted on CTS DynaSelect, as I mentioned before, using the DynaSelect isolation kit and the CTS detachable dynabeads CD4 kit. The input material contains 400 million CD4 T cells, and beads were added at 4:1 bead to CD4 positive cell ratio. The total process time was about 1 hour and 55 minutes for both target cell isolation and active release. And the flow data was analyzed on Attune NXT flow cytometer, and cells were counted on the nucleocounter NC3000. Here you see the phenotype of the cells in the starting material with an average CD4 positive cell frequency of 43%. And other cells, obviously, such as monocytes, B cells, and NK cells. After isolation, we achieve a 95% enrichment of target cells with very low frequency of contaminant cells present. After the target cells are isolated and the beads are released using the release buffer, the target cells in the output fraction are then counted and normalized to the number of target cells in the input material prior to isolation. This allows us to assess recovery. And as you can see here, we obtain high recovery of 88% on average. The data shows that cell viability is not negatively affected by isolation and release processes when compared to the viability of the starting material. As a matter of fact, the viability is slightly increased. that is most likely due to enrichment of viable cells during the washing steps in the DynaSelect protocol. We will continue this data presentation by showing performance data using CTS detachable dynabeads CD8 kit now. And the protocol for isolation of CD8 positive T cells from leukapheresis material is identical to the one for the CD4 T cells. So the following results are generated using the same scale of 400 million target cells and a B to cell ratio of 4:1. DynaSelect process time is still 1 hour and 55 minutes. This graph shows a comparison of cell phenotype in the starting material versus after isolation, and CD8 T cell fraction in the material, in the starting material, show an average frequency of 25%, and other cells such as monocytes, B cells, and NK cells are present. But after isolation, we achieve an average of 89% enrichment of the target cells with low non-target cell impurity. As for the CD4 T cell recovery results, the recovery obtained when using CTS detachable dynabeads CD8 kit combined with the release buffer was high, with an average of 88%. Cell viability remains high after DynaSelect isolation and release protocol, with an average of 94% frequency of viable cells at the end of the process, compared to 93% before. The detachable dynabeads CD4 and CD8 kits enable the user to run single subset isolations or combining the two products to obtain an output material containing all the CD4 and CD8 T cells from the leukapheresis in the starting material. So for this process, actually, it's identical to the two previous protocols presented, with the exception of using full culture leukopacks as a starting material and adding both detachable dynabeads to the same isolation bag. The process time is slightly higher to approximately 2 hours and 30 minutes, and that's due to the increase in scale. Purity of the isolated cells is incredibly high, 99% compared to the starting material, which is averaging 65%, while frequency of non-target cells is very, very low. Target cell recovery is calculated separately, and values of both CD4 and CD8 T cells recovery are high, showing an average of 94% for CD4 and 87% for CD8 T cells. Comparing viability before and after the DynaSelect isolation and release process shows an increased frequency of viable cells and the isolated cell fraction compared to the starting material. Again, we usually see that some dead cells are mostly washed away during the DynaSelect protocol, as I mentioned before. And these data here show the ratio of the CD4 to CD8 being very similar in the isolated material when compared to the starting material in the combined isolation release process. In summary, this is an overview of a growing platform of CTS detachable dynabeads that provide process flexibility, scalability, and consistent performance. I hope that I showed you today that our new CTS detachable dynabeads CD4 and CD8 kits achieve high purity and recovery with fast and automated process. And also that the CD4 and CD8 kits can be used either separately or in a combined protocol. Finally, I would like to thank you for your attention, and now I will hand you back to Caitlin.
Great, thank you, Haney, for a great presentation. We're now going to start our Q&A section. So just a reminder to our audience, please do keep your questions coming in. We will try to get to as many as we can during the Q&A session, and any we don't get to, we will get to by email. So the first question we have here for you, Haney, asks, what is the benefit of this product having VHH antibody fragments for both CD4 and CD8?
Yes, so there are several benefits. I mentioned a couple of them in the presentation. So the VHHs are produced in yeast, as I said, so they are animal origin free, and there is no risk now for these viral-like particles that are usually can be a problem with regular antibodies produced in hybridomas. That's kind of one advantage. Another advantage here, which relates to our release mechanism as well, because VHHs are very tiny, as I mentioned, and when they are placed or kind of conjugated to the bead and on a platform like the bead, together they have very high affinity, and now they're able to isolate the cells. But when they are released from the beads and now they are single VHHs, now they lose a bit of this affinity, allowing the VHHs now to be washed away from the cells. So that's kind of one of the main advantages.
Great, thank you so much for providing that insight. Our next question here asks, what is the recommended ratio of dynabead CD4 and CD8 beads to target T cells?
So for the detachable dynabeads CD4 and CD8, we recommend 4:1 bead to target cell ratio. And I'd like to clarify something here. When I mean by target cells, I mean cells that express, obviously, CD4 or CD8. I would also like to point out something that might help in the process, which is that there is no need to phenotype the starting material by flow cytometry before starting of isolation. We routinely use the COA of the leukopak and the information present there to calculate and determine the bead volumes before isolation. So it's not necessary to do a flow phenotype analysis prior to isolation.
Great, that's good to know, I guess. So our next question here asks, can I try out the CTS detachable dynabead CD4 and CD8, but do so manually with columns and not with CTS DynaSelect magnetic separation system?
So all of our detachable dynabeads products, they're intended to be used together with DynaSelect magnetic separation system. So they are developed together and they are optimized with each other. So while small magnets can be used to assess functionality of the product in a laboratory scale, it does not allow the user to see the full benefit or accurate performance of these products. So testing our CTS detachable dynabeads without the CTS DynaSelect system will not result in the level of yield and purity that I showed today.
Great, thank you so much for answering that question. Our next audience member asks, can you share more on the method and principle for detachment?
Yes, so as the figure kind of suggests in my presentation, so the beads are conjugated with these VHHs that bind to the target cells. And we have another reagent that we use to add to the bead-bound cells. And this reagent will detach the bead from the VHHs that are now attached to the cells. And as when the beads are detached from the VHHs, now the VHHs are single molecules on the cell, and they are washed away easily from the cell surface.
Great, thank you. And that was a very insightful answer. So our next audience member here has kept it short and sweet. They simply asked, how was viability assessed?
Viability assessed by flow cytometry. So we have a flow panel that includes several antibodies for phenotype analysis, and we have a viability dye that is included in the flow panel.
Great, thank you. Our next question here asks, is there any difference in avidity of the new antibody, anti-CD3, anti-CD8, in the detachable reagent as compared to the prior CTS bead reagent?
So when we're talking about avidity here, I am assuming you mentioned the capacity of the antibody to bind to several molecules. And so obviously the VHHs are small, kind of single fragments, and by themselves, separate, they would not have high avidity. But in our case, we coat the beads with lots of VHHs, and that increases the avidity, obviously, because now we have lots of binding sites on the bead that can bind to the cells. So that is the genius of this mechanism, that the VHHs are on the beads, they have very high avidity, and they are able to bind to the cells, and also high affinity as well. But then when the bead is released, then the VHHs can be washed away.
Great, thank you for providing that insight. Sort of as a follow-on, I guess the previous question is, this user asks, how do you calculate your recovery efficiency?
Yes, so when we talk about recovery here, what we do is we calculate the number of target cells in the starting material. That depends on the scale. So I presented today 400 million target cell scales and a quarter leukopak scales, where you can also be calculated either through the COA data or taking a sample and phenotype that on flow afterwards. And then we get a number of these target cells in the starting material, and then we look at the number of the target cells in the positive fraction at the end of the isolation. And we simply just divide these two numbers together and get a percentage of cells that we recover afterwards.
Excellent, thank you. So this next question has two parts to it. The first part asks, can you co-select CD4 and CD8 in a single step? And then the second part, I guess pertains to the first part, says, are there any residual CD4 and CD8 beads remaining?
Yes, so the first part is yes, you can, as I showed today as well, you could use the kits separately and isolate only CD4 cells or only CD8 cells, but you can combine the beads together and isolate both CD4 and CD8 at the same time. So that the answer to that part of the question is yes. And when it comes to residual beads, so DynaSelect comes with bead removal kit that is extremely efficient in removing beads from the cells, and we regularly obtain way below the standard 100 beads per 3 million cells standard that is accepted in the industry. And when I say way below, I mean in sometimes 1 or 2 or even some these type of numbers.
Great, thank you for that. Our next user here, or audience member here, asks for something specific to the platform. They asked, are the detachable dynabeads compatible for use in manual processes and or with other platforms, or only with the DynaSelect?
So as I mentioned, all the data presented and all of our kind of know-how and work around that is in conjunction with DynaSelect. And we developed these products in parallel together. So they are extremely optimized together and work perfectly together. So there is, I can't speak to how the performance of these products would be if they are not used together with DynaSelect. But say, as well, by saying that, I could also say that it is possible to do manual protocols with these beads on small magnets and get some kind of idea of how the beads perform, but this is not translatable at all to how the performance is on DynaSelect.
Great, fantastic. So this next question is a follow-on to the co-select question. This audience member asked, in the combined isolation release process, the ratio is still 4:1? Do you add equal parts of anti-CD4 and anti-CD8 beads to reach this ratio?
No, we don't do that because the ratio of CD4 to CD8 in the starting material is not 1:1. So the process is to identify the ratio of the CD4 and CD8 in the starting material, and then adjust the volume of the beads to that to a 4:1 ratio to each kind of cell type. So you do very similar calculation of the volume of the beads as you do with a single isolation, but in that case together in one protocol at the same time.
Great, thank you. And thank you for answering all those questions today. Unfortunately, that is all we've got time for today. As I mentioned previously, any questions we didn't get to during the live session, we will answer by email. The webinar will also be available on demand from tomorrow. So please do look out for an email from us with that link. And all that's left to do is to, of course, thank Haney once more for a great presentation. And of course, thank you to the audience for listening. We hope you'll join us again soon.
Thank you very much.